Hello, my name is Andy Kim. It's my pleasure to speak to you today on accelerated tech transfer for COVID-19 programs at Samsung Biologics. First, I'll start with a brief introduction to SBL's facilities here in Korea. Then we'll look at some expedited tech transfer timelines for 2 COVID-19 programs. We'll take a deeper look at how we achieve that acceleration, and then we'll wrap up with how we address some unique COVID-19 challenges and lessons learned. Here in Korea, we have 3 plants on our site with a 4th plant under construction. My focus is on plant 3, where my team is based. We support tech transfers into this facility, which has 12 15,000 liter bioreactors. We have a track record of transferring across many different scales. And for the 2 programs I'll talk about in the next several slides, they fall into a category of transferring from about a 2,000 liter scale on the client end to 15,000 liters in the aforementioned plant 3. And this is all supported by additional work sometimes performed in our MSAT labs, where we typically use 10 liter bioreactors. So a typical timeline for tech transfer spans 6 months from kickoff to vial thaw for an engineering run. In the 2 examples described on this slide, we completed tech transfer within a 3 to 5 month window. And in both cases, we bypassed our usual engineering run and went straight into a clinical or PPQ campaign. This was achieved by several means, including performing activities in parallel. So a good example of that is our lab scale verification run, which we normally perform a couple months ahead of the engineering run. But in these 2 programs, as you can see on the slide, the lab scale verification runs were performed in an overlapping schedule with our manufacturing campaign. So in order to accelerate tech transfer, we didn't introduce some revolutionary new idea, but instead we relied on solid execution of the fundamentals and focusing on those areas where we can accelerate by realizing time savings and mitigating risk. So first, for these programs and for any tech transfer project, careful planning and clear communication is really important, especially on our end, the receiving side, because we drive so many of the activities that feed into our manufacturing campaign. In order to accelerate, we really benefited from working with a platform manufacturing process for our antibodies. And at the beginning of our projects, we agreed upfront with our partners that traditional timelines may not work. We'd have to get creative because you can't have your cake and eat it too. When time is short, we have to think about where we can save time. And I've mentioned parallel activities. We approached our work in a phase-structured approach, and underlying the entire workflow from beginning to end, we really emphasized the early assessment, early identification of critical gaps, continuously monitoring them and mitigating the risk. So clear communication is paramount. When you have misunderstandings, that leads to delays and rework, and time is of the essence. So on the sending side, what really helped for some of these projects was when a client assigned an individual to serve as a single point of contact who could help manage the document reviews and ensure nothing falls through the cracks. On our end, even before tech transfer started, we thought about how can we ensure smooth communication. So we assigned technical stewards or technical leads who not only had oversight of all the technical pieces, but who could serve as a clear decision maker and make sure that the programs move forward. So they served to bridge the cultural gap between Korean culture and Western culture. The technical stewards also were put into place to help address any language challenges. So English is the primary business language used at SBL, and we have a pretty high capability, we think. But the capability does vary widely across our many personnel. So not only did we have technical stewards in place, but our project meetings were facilitated by personnel who have much stronger English skills, and any decisions were confirmed via the written word in order to prevent any misunderstanding. We have to acknowledge that we really benefited from working with an established antibody manufacturing platform. By having a single paradigm, we can really expect consistent and reliable scale. So even before we receive any process details, we kind of know what to expect in terms of likely technical challenges and what are the most critical parameters. This, coupled with leveraging any historical data or understanding from the sending side for their platform process, really allowed us to focus our precious resources on the critical gaps, on the items that require the most attention. To accelerate the timeline, we had to come together and discuss and decide redefine what is the acceptable level of risk. So for example, for these two programs, we performed tech transfer in parallel with an accelerated process development timeline. So we had to think about where do we have room to give and what's fixed. There's no compromise. So one thing that was fixed for us at least was we made clear when defining the project timeline, we really need process lock at least 1 month ahead of our manufacturing campaign. If there's any delays to process lock, then it would result in a delay to the campaign. On the other hand, thanks to a high level of engagement and transparency on the side of the sender, we received frequent updates on the development process, progress on their timeline, on their results, and this allowed us to give feedback in real time and to assess the manufacturability of their proposed ranges. We could then suggest any process modifications and working together, and the client on the sending side could evaluate any proposed modifications in their development work. So for example, one case that stands out is due to vessel volume constraints, we asked to look at a higher UF1 target concentration to be used in our manufacturing campaign. We also incurred additional risk by eliminating the engineering run for the schedule. So we had to agree together upfront that performance may not be the best, and we have to accept performance trade-offs. So in one case, we saw much lower titer than had been projected. And in other cases, we saw some transfer losses of the proteinase step that were easy to fix, but require time. And time is something we didn't have during the campaign. We just had to live with these. But that was okay because the product quality was good. Normally going into a campaign, we require a high level of confidence in the process. But without an engineering run, we sought to mitigate this risk by seeking to achieve the right level, an acceptable level of product quality through our small-scale verification runs. We also thought about where can we devote additional resources or build flexibility into our timeline to where we think we need it the most. And so a good example of that is for our PPQ campaigns. We asked for a larger number of PPQ batches in our schedule so that if we see any non-process related issues in the first couple batches that are easy to fix, we'd still have a sufficient number of PPQ batches to demonstrate consistent production and control. And then lastly, this example doesn't apply to these two programs, but it did come into play for some other COVID projects we have, where we did schedule an engineering run in the manufacturing schedule, but there was a very short time window between that engineering run and the subsequent campaign. So in those cases, we mitigated risk by checking the product quality through our concurrent satellite runs performed at small scale. We also saved time by agreeing upfront that we would limit process improvements to critical fixes, to those issues with a clear root cause and CAPAs. And then we also aligned on the document revision timeline. So that way, when it came time to revise the documents, we could focus on the content, what needed our attention, and not waste time on figuring out the details for when we would start review or have a document ready and done by. So in terms of thinking about a phased approach and orchestrating these complicated parallel activities, that requires a very clear understanding of the key deliverables, the key milestones. And so for us, when we do our tech transfer, everything starts with the client documentation, their process description. This feeds into our process description, our control strategy, which then feeds into and informs our manufacturing batch records for the plant. So at the beginning of the project, for both these cases, we asked, please share with us, please send us your documents as soon as possible. But when those documents were not available, such as a process description, one way we mitigated the impact was by completing the documentation in a rolling format so that we could get the most urgent, the most time-critical information as soon as possible. On our end, we thought about how can we smooth communication when it came to dealing with documents and many complex activities. So we generated a document flowchart to visualize the process. And so I show a copy of that on the slide, which it's really hard to read, but that's okay because my point is actually that it's complicated. So by generating this tool, we can easily communicate what documents we plan to write, what is a purpose, and how do they all connect to each other. And that helped us to get onto the same page. Looking at the bottom half of this slide, you'll see just a generic timeline for tech transfer from kickoff to the actual campaign. There's just a couple points I want to emphasize or re-emphasize. One is throughout the process, from beginning to end, we continuously monitor the risk. We mitigate risk, respond to it as quickly as possible, and we were always reassessing, is there anything we've missed? When it came to our documents, both on the sending side and on the receiving end, we sometimes author documents in a rolling format. So a specific example is in the case of upstream, which is on the critical path. For the upstream process description, maybe version 1, describe the process only up to the shake flask expansion. And then maybe version 2, which followed a week or two later, would then describe the operational details for the bioreactor step. And then lastly on this slide, just addressing supply gaps. At the beginning of our work, we prioritized long lead items, such as virus filters, things that are going to take a lot of time. We then focused our attention on less risky items. And then we followed up with the least riskiest, the more common items such as 0.2 micron filters or common chemicals like sodium chloride. In those rare cases where we could not source the supply we needed, we then explored whether the client on the sending side could send these materials to us through drop shipments. So from week 1, we sought to rapidly identify any facility fit issues and supply gaps. So the first on our list of priorities was CapEx issues, equipment issues, because these have a long lead time. When we had any CapEx gaps, we first sought to address these by transferring available equipment within the SPL site across the different plants. We also, when that failed, we sought to explore if the client could drop ship spare equipment to us in order to meet our manufacturing timelines. In those cases where we did have equipment available, but it wasn't the perfect fit, we mitigated risk by performing wet runs in the manufacturing suite in order to fully assess the risk involved. So a good example is for the UF operation, exploring the use of larger volume mobile vessels for the initial concentration steps due to the limitations on our escape retentive tank. After CapEx items, we then focused on raw materials and consumables because those normally have about a 4 to 6 month lead time for us. In doing our mass balance calculations, we assumed worst case quantities for production, and an example of a variable titer range where we're not sure what titer we're going to get, we planned for the worst case, which was the highest titer that we expected. And that way we could fully process forward any material that was generated by our bioreactors. Second, when we identified any high-risk items, we mitigated risk by recommending alternatives for evaluation. So a good example, again, is the virus filters. There's a lack of vPro supply. And so in this case, we recommended some alternatives that were easier for us to source, which the client then took back and assessed in their development studies, which enabled us in the end to use these alternatives in our manufacturing campaign. Lastly, we would normally execute a manufacturing campaign with a sufficient amount of safety stock for all our critical materials. But because of the ongoing supply issues associated with COVID-19, we had to live with the fact that there are just some items for which we have little to no safety stock. So if there are any issues, there are no backup batches because there's no backup materials. And then after CapEx and raw materials, we focused on potential manufacturing issues. And this is something that we continuously reassess throughout our workflow. So in the present COVID environment, face-to-face interactions and international travel is really limited. So for these programs, from start to end, all of our meetings were held virtually using Zoom or an appropriate tool. Additionally, when it came to actual production in our plant, normally we'd expect the sending site to send an SME, someone to be present on the floor during production. However, since that wasn't available to us, we mitigated the risk by agreeing upon 24-hour accessibility of an SME on their end who could respond through phone or by message or by email, but who would be available so that we could respond quickly to any manufacturing issues. So to wrap up, I hope these slides and this information helps you to understand how we were able to successfully execute these COVID-19 tech transfer projects within a very small time frame of 3 to 5 months in order to meet an urgent medical need. This was only made possible first and foremost by a strong partnership between the sending side and our clients and the receiving side, coupled with careful planning and clear communication. Throughout the process from the very beginning, we emphasized the identification of the critical path items, and we sought to mitigate risk and continuously reassess that and monitor that. So through proper planning and our various mitigation measures, we were able to set ourselves up for success in our manufacturing campaigns, even though the time was so short. But if confronted in a future health crisis, our best recommendation is to start tech transfer as soon as possible. Because even with limited resources, even with an undefined process, the sooner you start, the more time you will save. So with that, I'd like to thank you for your attention, as well as to acknowledge the many individuals, too many to list here, who made all of this possible. Thank you.